Welcome to the Deep Dive. Today we are going deep into vet neurology.
Yeah.
With a case study.
Oh, it's fascinating.
That is both fascinating and incredibly rare.
Incredibly rare.
We are talking about a spinal cord tumor in a dog.
Okay.
A ganglioglioma.
The ganglioglioma.
And it is so rare.
In
fact that this case, straight from the Veterinary Radiology and Ultrasound Journal,
yes,
is the first of its kind.
Yeah.
Confirmed using a very specialized technique.
That's really, it's a real testament to how advanced veterinary medicine has become.
Right.
We're going to break down this complex case of all the technical details, but don't worry, we'll make sure you understand all the key takeaways, even if you're not a veterinarian.
Awesome. So let's meet our patient.
Okay.
A 7.5-year-old German Shepherd.
Okay.
Who started showing some worrying signs.
Right.
Dragging his left forelimb, difficulty walking.
Yeah.
And eventually not even putting weight on that leg.
Oh.
It sounds pretty straightforward, right? A classic limp.
You'd think so, wouldn't you?
Yeah.
But this wasn't just a simple limp.
Oh.
The dog was also showing signs of ataxia in his hind limbs.
Okay.
Meaning his coordination was off.
Wow.
And get this.
Okay.
There was noticeable muscle atrophy.
Oh, my God.
And asymmetry.
Okay.
Even in his facial muscles.
Whoa. Now, that's where it gets interesting.
Yeah.
So we're talking about a neurological issue.
Yes.
Not just a localized problem in the leg.
Exactly.
What happened next? Would they figure out what was going on right away?
Not even close.
Oh, no.
Here's where the mystery deepens.
Okay.
The vets ran the usual suspects.
Right.
X-rays. Yep. Blood work. Even a full-body CT scan.
Wow.
And everything came back normal.
What?
Talk about a head scratcher.
That's incredible. So if all the standard tests were clear,
right,
how on earth did they find the culprit?
Well, they finally turned to MRI, specifically of the head and cervical spine. And there it was, hiding in plain sight.
Oh, wow.
A 4-centimeter-long mass.
Oh, my gosh.
Right in the C6, C7 region of the spinal cord.
Okay, so they found this mass on the MRI.
Yes.
What did it look like? Did it scream tumor right away?
It wasn't exactly shouting, but it definitely whispered suspicious.
Okay.
The way the mass appeared on the MRI with those technical terms like T2 weighted hyperintense and T1 weighted isointense gave the vets some strong clues.
Okay.
Think of it this way. Right. Imagine shining different colored lights on the mass.
Okay.
The way it reflects each color tells us something about its composition.
Oh, that makes sense.
Oh.
So it was the specific pattern of light and shadow on the MRI that raised a red flag.
Exactly.
But how did they jump from this mysterious mass to suspecting it was a ganglioglioma?
Right.
This super rare type of tumor.
That's a great question.
Yeah.
It's a combination of experience. Knowing the typical locations and appearances of different spinal cord lesions.
Right.
And a healthy dose of intuition.
Okay.
The location of the mass, its size. Yeah. And the way it behaved on the MRI all pointed towards a ganglioglioma.
Okay.
Even though it's incredibly rare in the spinal cord.
So they had their suspicions.
They did.
But how did they confirm it was actually a ganglioglioma?
Right.
And what exactly is a ganglioglioma anyway?
Let's unpack that.
Okay.
A ganglioglioma is a type of tumor that's made up of two main cell types.
All right.
Glial cells, which are the support cells of the nervous system, and neurons, which are the nerve cells responsible for transmitting signals.
Got it.
What makes gangliogliomas unique is that they contain both of these cell types.
Interesting.
And the neurons within them are often abnormal, or what we call dysmorphic.
So it's like a mixed up tumor with both support cells and nerve cells all jumbled together.
Exactly. And to make things even more complicated, they're typically slow growing and often don't cause any symptoms until they reach a significant size.
So in this case, they found this mass in the dog's spinal cord. Right. And suspected it was a ganglioglioma. But how did they know for sure?
So in this particular case, surgery wasn't an option for the dog. Due to the location and size of the tumor.
Right.
And to confirm the diagnosis, they needed to examine the actual tissue under a microscope.
So they had to wait until...
Yes. Sadly, the dog was eventually euthanized due to his declining condition.
Oh, that's sad.
But this allowed them to perform a necropsy, basically an autopsy for animals, and get a closer look at the mass.
I see.
Yeah.
It's heartbreaking, but it also gave them the opportunity to confirm their suspicions.
Yes.
And learn more about this rare condition.
Exactly.
So what did they find during the necropsy?
Well, when they opened up the spinal cord, they found a tan-colored, soft swelling right in the C6, C7 segment, just as the MRI has shown, swelling. About 4 centimeters long, had completely distorted the normal structure of the spinal cord.
Wow.
But to really understand what they were dealing with, they needed to go deeper, literally into the microscopic world.
That's where things get really fascinating, right? Taking a peek at the cellular level to see what's really going on.
Precisely.
Cool.
And that's where we'll pick up in the next part of our deep dive. We'll explore the microscopic findings that definitively confirm the diagnosis of a ganglioglioma.
I'm already hooked.
Great.
We'll be back in a flash to delve into the microscopic details and uncover what makes this case so groundbreaking.
Awesome.
Stay tuned.
Yeah. Welcome back to the deep dive. Before the break, we were talking about that crucial moment in the case where they finally got their hands on the actual tumor tissue during the necropsy.
Right. It's like they were finally able to piece together the physical evidence in this medical mystery. So what happened when they put that tumor tissue under the microscope?
This is where the real detective work began. Under the microscope, the tumor showed a fascinating mix of cells. Remember those 2 key players in a ganglioglioma? Glial cells and neurons?
Yes.
Well, there they were right in front of their eyes.
No way.
They saw neoplastic glial cells. The support cells gone a bit haywire. And mature neurons, the nerve cells themselves.
So they actually saw the 2 different cell types that make up a ganglioglioma. That's pretty cool.
It is, but it gets even more interesting. The neurons weren't quite normal. They were larger than usual. Oddly shaped. Had some abnormalities in their internal structures. We call these dysmorphic neurons. And they're a hallmark feature of ganglioglioma.
Dysmorphic neurons?
Yeah.
Like misshapen brain cells? Okay, I'm trying to picture this. Was there anything else they saw under the microscope?
They sure did. Alongside those dysmorphic neurons, they found a few other interesting characters.
Oh, really?
There were some Rosenthal fibers. These long, brightly staining structures, often associated with slow-growing tumors.
Got it.
And they also spotted some eosinophilic granular bodies.
Eosinophilic granular bodies.
Round, pink staining structures that are thought to be remnants of degenerating cells.
Okay, I'm starting to get a sense of the complexity here. So many different cell types and structures all jumbled together. But they weren't content with just looking at the tumor under a regular microscope, right? They had to bring out the big guns.
You're right. Remember that powerful technique we mentioned earlier? Immunohistochemistry, or IHC for short.
Right, IHC.
Yes.
It's like using different colored spotlights to identify actors on a stage.
That's a perfect analogy. In Champ's case, they used specific antibodies like tiny guided missiles to target particular proteins found in different types of cells. These antibodies were linked to a dye that glows under the microscope, illuminating the specific cells they were attached to.
So they basically stained the tumor with these special antibodies to see which cells lit up.
Exactly. To highlight the neurons in the tumor, they use an antibody against a protein called neuin, which is found primarily in neurons. And as expected, the neurons in the tumor lit up brightly, confirming their presence.
That's so cool. So they spotlighted the neurons. What about the glial cells? How do they confirm those were there?
For the glial cells, they used an antibody against GFP, or glial fibrillary acidic protein. Another specific marker for these cells. And as expected, the glial cells in the tumor also stained positive, confirming the mixed nature of the tumor.
So IHC provided that double confirmation, showing a mix of both neurons and glial cells, just like you'd expect at a ganglioglioma.
Case closed, right?
Well, almost. They had one more trick up their sleeve.
What?
A technique that allowed them to zoom in even closer.
To see the tumor at an incredibly small scale.
Wait, there's more. Tell me they didn't pull out another high-tech gadget. What could be more detailed than IHC?
Hold on tight, because they brought out the big guns.
Okay.
Transmission electron microscopy, or TEM.
TEM. That's the one that lets you see things at an incredibly small scale, right?
Yeah.
Like way smaller than what you can see with a regular microscope.
You got it. TEM uses a beam of electrons to create an image of a sample, allowing us to see structures down to the nanometer level. Think of it as the ultimate zoom lens for the microscopic world.
Okay, I'm picturing it. So what did they see when they zoomed in on Champ's tumor with TEM?
Remember those dense core granules we mentioned earlier? The ones that are a hallmark feature of gangliogliomas?
Yes, I remember. They were found in the neurons.
Well, with Tim, they were able to see these granules in exquisite detail.
Okay.
Numerous dense core granules, all clustered within the neurons of the tumor. It was the final piece of the puzzle.
Wow.
The definitive confirmation that they were dealing with a ganglioglioma.
Wow, that's incredible. It's like they went from a blurry image to a high-definition picture, all thanks to Tim. So they had all the evidence, the clinical signs, the MRI findings, the microscopic appearance, the IHC results, and finally the TEM confirmation. It's like they built an airtight case.
Exactly. And here's the kicker.
Yeah.
This is the very first reported case of a canine ganglioglioma confirmed by TEM. It's a significant finding not just for this particular dog, but for the entire field of veterinary neurology.
That's amazing. But I have to ask, what does all this mean for other dogs? Does this case change anything in how we approach spinal cord tumors in dogs?
That's the burning question, isn't it? And that's what we'll be discussing in the final part of our deep dive.
I can't wait. We'll be back in a moment to explore the implications of this groundbreaking case. Stay with us. Welcome back to the deep dive. We've been on quite a journey with this case, haven't we?
We have.
From puzzling symptoms to high-tech microscopes, it's been a whirlwind of information.
It really highlights the complexity and the incredible advancements in veterinary neurology.
Yeah.
But remember, it all started with one dog
and
a team of dedicated veterinarians determined to find answers.
Absolutely. And before the break, you mentioned that this was the first reported case of a canine ganglioglioma confirmed by TEM.
It was.
That's huge. What does this mean for the future of diagnosing and treating these tumors in dogs?
Well, it's a significant step forward. Having this ultrastructural confirmation adds another layer of certainty to our diagnoses. It's like having a high-definition image to back up what we see under a regular microscope.
So it's not just about confirming the diagnosis in this one case. It's about having more confidence in our diagnostic tools and techniques moving forward.
Precisely. And while TEM is a very specialized technique, not readily available in all veterinary settings. This case demonstrates its potential value in these challenging and rare situations.
It also underscores the importance of collaboration. It took a team of experts, clinicians, pathologists, radiologists, and researchers to piece together this diagnostic puzzle.
Absolutely. It's a beautiful example of how different disciplines can work together to advance our understanding and ultimately provide the best possible care for our animal patients.
So this case has certainly pushed the boundaries of our knowledge about gangliogliomas in dogs. But I'm curious, what are the practical implications? Does this change anything for vets on the front lines, so to speak?
Well, for one, it raises awareness about the possibility of gangliogliomas in dogs. Even though they are extremely rare. Vets should be aware of this as a potential diagnosis, especially when faced with unusual neurological symptoms that don't seem to fit the usual patterns.
It's a reminder to keep an open mind and consider all possibilities. Especially when dealing with neurological issues, which can be so tricky to diagnose.
Exactly. And early detection is key. As it is with most medical conditions, the earlier we can diagnose a spinal cord tumor, the better the chances of successful treatment, whether that's surgery, radiation, or other therapies.
But we still have so much to learn about gangliogliomas in dogs.
We do.
What causes them?
Are
there any genetic factors involved? Are there ways to prevent them altogether?
Those are all excellent questions, and unfortunately, we don't have all the answers yet. This case highlights the need for further research in this area.
It's like opening a new chapter in veterinary neurology.
It is.
We've taken a giant leap forward with this case, but there's still a whole uncharted territory to explore.
You hit the nail on the head. And who knows what other groundbreaking discoveries await us as we continue to delve deeper into the complexities of animal health.
It's exciting to think about all the possibilities. This case, though, a sad one, has paved the way for a deeper understanding of gangliogliomas in dogs.
It has. It's a reminder that every case, no matter how rare or challenging, has the potential to teach us something valuable and contribute to the larger body of knowledge.
And that knowledge, in turn, can help us better diagnose, treat, and hopefully one day prevent these conditions in other animals. That's what makes veterinary medicine so rewarding, isn't it? The constant pursuit of knowledge and the drive to improve the lives of our animal companions.
It certainly is, and it's a reminder that even in the face of challenging diseases, there's always hope. And there are dedicated individuals working tirelessly to make a difference.
Well said. This deep dive has been an incredible journey, and I think we've covered some truly groundbreaking stuff today. It's been fascinating to explore the world of veterinary neurology through the lens of this rare case from Vet NeuroJar. Keep those minds-inspired hearts light and tails wagging.